There's certain knowledge and skills more likely to be assessed in A-level biology than others. So whether you're about to sit your exams or just starting your A-level biology journey, it is essential you are familiar with the apparatus and techniques checklist that all exam boards use to ensure students are familiar with common instrumentation, apparatus and skills relevant to the course. First, we will go through the list. Then I will tell you from my analysis which comes up most often. And I can tell you from analysing AQA papers between 2017 and 2025 that typically 6 come up in some way, shape, or form across the 3 exam papers. 6 out of 12. That is a good hit rate. So you must use this list to help you prepare for exams or mock exams. You'll develop many of these skills across your required practicals across the course. However, the examiners will include 15% of marks asking you to show knowledge of the practical skills that you've developed. So let's work through our list. First of all, ATA is the appropriate use of apparatus to record a range of quantitative measurements. There are some examples of measurements and equipment shown here, and you should be able to select the most appropriate equipment to aid accuracy and precision. You need to know how to use them to avoid systematic or random errors, and you need to understand the associated uncertainties. You will also need to perform mathematical calculations using the uncertainties of the measuring equipment. Now, I'm not going to be able to tell you everything you need to know about each of the skills and apparatus in this video, so check out my other videos on the channel and my playlist for the apparatus and techniques short series if you want to know more. The links are in the description. ATB involves using instrumentation to record quantitative measurements. The examples listed include colorimeters and potometers. However, some other common ones that come up include calorimeters and respirometers, both of those more on paper 2, but also hemocytometers. Make sure you know how they work and key parts of the method to ensure accurate results. ATC focuses on the use of laboratory glassware. Now, you should be familiar with equipment such as pipettes, burettes, volumetric flasks, and measuring cylinders. One important technique is carrying out serial dilutions, and you might be doing this to prepare solutions of known concentration or dilute a cell culture to allow more accurate determination of its concentration. You might need to describe how to perform the serial dilutions in an exam question or perform calculations using the equation C1 times V1 equals C2 times V2. And remember, you don't get given the equations, so for more advice on which maths equations you need to memorise for biology, check out my video in the description. ATD is the use of a light microscope at both low and high power, including the use of an eyepiece graticule and a stage micrometer. You should be able to calculate magnification, determine actual size, and describe how to calibrate the eyepiece graticule using the stage micrometer. Some other maths you might need to do relating to this skill is calculating stomatal density. Again, check out the video below for more guidance on the maths. ATE requires you to produce scientific drawings from observations with annotations. And there are some fundamental rules for scientific drawings, and you need to know how a tissue map may differ from other drawings. The most common error is not putting a scale bar or magnification on drawings, or the drawing not actually looking like what you are seeing. Scientific drawings should be clear, use single continuous lines, avoid shading, and include appropriate labels and magnification where possible. ATF involves using qualitative reagents to identify biological molecules. You should know the tests for reducing sugars, non-reducing sugars, starch, proteins, and lipids, and include the expected positive results. And make sure you can describe the colour change from and to for an exam question. For the lipid test, you must describe the positive result as a milky emulsion. You cannot say it goes white. For the Benedict's test, make sure you know how to turn qualitative data into quantitative data by either using a colorimeter or drying and weighing the mass of the precipitate formed. ATG covers the separation of biological compounds using chromatography or electrophoresis. Now, thin layer chromatography can be used to separate photosynthetic pigments and is often completed as a required practical. You will need to do the maths for calculating the RF values also. Gel electrophoresis is commonly used to separate DNA fragments according to size and is learned about in the gene technology chapters. If you are lucky enough to have Amgen project running in your school, you'll get some hands-on experience using electrophoresis to perform your own DNA fingerprinting, and I highly recommend it. ATH focuses on the safe and ethical use of organisms to investigate responses and physiological functions. Several required practicals involve living organisms, including at least one dissection, so make sure you can clearly describe how to work safely and ethically. ATI involves microbiological aseptic techniques, and you will complete or have completed a required practical investigating antibiotic resistance and working safely with agar plates and broth cultures whilst minimizing contamination through sterilization and careful handling procedures. Now, for exam questions, make sure you look out for the command word describe or explain. For explain, you must justify the purpose of each part of the technique. ATJ is the safe use of instruments for dissection. You will write or have written many risk assessments across the course, so be prepared to write one in the exams. Now, ATK covers sampling techniques used in fieldwork, and this includes quadrats, line transects, belt transects, sweep nets, pooters, and mark release recapture methods. These techniques are used to estimate abundance and distribution of organisms. So make sure you know which method you would choose for different types of organisms or different aims of the investigation. You must be able to write a method and perform calculations, which I have other support videos on. Finally on the list, ATL, and this involves the use of ICT, and this includes data loggers, computer modelling, or software for data processing and analysis. Now, in an exam, the only software you are going to be using is your calculator, so this one doesn't really count. Now, when I say typically 6 of these 12 skills come up across the exams, I didn't include using a calculator as one of these 6. 10% of marks is maths, so of course this one comes up. That means 6 of the other 11 typically come up across the 3 exam papers in either short or longer answer questions. So, which of these techniques come up the most? Here is my analysis of the last 9 years' papers for AQA. And looking at this table, there are 3 practical skills that jump out as being assessed at high frequency. Firstly, B, using appropriate instrumentation to record quantitative measurements. Now, within the different types of instrumentation, the most common technique appearing was the colorimeter, and this was 5 times, often for calibration curves, sometimes for enzyme rate of reaction. Now, the other named piece of equipment on the tracker is the pedometer, but that didn't actually come up that much. The respirometer, however, has come up 4 times. All of these were on paper 2 or 3. So if you're revising paper 1, revise colorimeters, calibration curves, and pedometers. If you are revising paper 2, revise respirometers, calorimeters, and colorimeters. If you're revising paper 3, revise all of these. Next, let's look at D, use of the light microscope at high power and low power, including use of the graticule. Now, there's so many ways they can test your understanding of microscope skills. Unsurprisingly, it has come up a lot. So when you are revising, make sure you practice your microscope calculations and you practice interpreting electron microscope images. Examiners love these. Know the importance of steps when making slides, especially for CPAC 2, and be able to describe how you use the microscope, including how you use your stage micrometer. Now, the third of these skills that comes up a lot is sampling organisms in fieldwork, probably because it's covered in chapters 10 and 19. And this means it's an important topic to include for revision on papers 1, 2, and 3. So I hope you can see how important this list is to include in your exam preparations. As you go through your papers in the summer, you should consider which have come up already on papers 1 and 2 when you are preparing for paper 3. Thanks for listening, and do check out more helpful videos and downloads in the video description. And if you found the video helpful, do share with fellow classmates.